we focused on the differences between B3-leap, which has been considered also in previous work the best compromise for the study of astrochemically relevant reactions, and CAM-B3-leap, which is presented as an improvement of B3-leap considering the long-range interaction. In all cases, a couple cluster single-point calculations have been performed considering the geometries optimized at different level of theory in order to understand the variation of the energy at couple cluster level. Previous informations are available for the reaction between oxygen and acrylonitrile, both from a theoretical and an experimental point of view. In particular, a theoretical investigation of the triplet surface showed two possible reaction mechanisms. One of them is a nitrogen abstraction process leading to the formation of the OH radical together with the cofragment, which can be CH, CHCN, or CH2, CCN, depending on the different isomerization. And the other one is the formation of the long-range interacting complex, which can then lead to the formation of covalent bond intermediates, and different isomerization steps can lead to the formation of several products, including hydrogen with the related cofragments, but also formaldehyde, NCO, CO, and so on. I can report here some results I obtained during my work. In particular, here I reported the geometry, so the bond distances in angstrom and the angles in degree that I obtained with different methods, in particular B3-leap and CAM-B3-leap in blue and green respectively. And I reported also the B2-leap D3 results in order to have a comparison with a functional which is considered a good way to study long-range interaction, including also the dispersion. As we can see, there are no particular differences in the value of bond distances and angle obtained with B3-leap and CAM-B3-leap for what concerns the covalent bonds, carbon-carbon, but also carbon-nitrogen and carbon-hydrogen, while some also relevant discrepancies can be observed in the analysis of the long-range interaction, where, as example, the distances between the oxygen atom and the different atoms of the partner molecules. And what we can see is that the CAM-B3-leap, there are differences between B3-leap and CAM-B3-leap, and the CAM-B3-leap results are more in line with the B2-leap D3 results, both for the bond distances and for the angles. Once we obtain the energies and the harmonic vibrational frequencies in order to be sure that the intermediate point that we found is a minimum point since all the frequencies are real, we performed an analysis of the energy. And here I reported for the same stationary point for the long-range complex, both the energy obtained with different methods, and on the second columns there are the energy, the relative energy with respect to the reactant energy asymptote obtained at the couple cluster level, considering the geometry optimized with the different methods shown in the table. From the results, we can see that the energy obtained with the different methods is very different, and the difference in energy is in some cases is also higher than the uncertainty related to accurate calculations, which can be considered around 5 kJ per mole. While if we move to the couple cluster relative energy, we notice a good agreement between all the different methods, between the energy obtained by analyzing the geometry optimized with different methods. And in particular, we can see very, very small difference between the couple cluster energy obtained with the B3-leap and CAM-B3-leap optimized geometry. So once we noticed that there are basically no difference in the couple cluster energy, we decided to continue with the study of the first step of the reaction. Here I reported the very first stages of the evolution of the reaction. Also in this case, the energy are couple cluster energies obtained with the geometries optimized with the three methods reported, B3-leap, CAM-B3-leap, and B2-leap D3. As we can see, once the long-range interacting complex is formed, the reaction can proceed through the formation of a covalent bond intermediate. We have to say that the formation of a long-range interacting complex is a very important point for most of the biomolecular reactions, because it's believed to be present in most of them, and it can enhance or hinder the reactivity of the species, depending on the presence of the transition state, if it's higher or lower than the reactant energy asymptote. In this case, the transition state that we identified, in which the oxygen interact with the plane of the acrylonitrile molecule, is located 4 kJ per mole below the reactant energy asymptote. So in principle, the reaction can be important and can be feasible also considering harsh conditions of temperature and pressure, which are typical of some regions of the interstellar medium, as example. The transition state leads to the formation of a covalent bond intermediate in which a new carbon-oxygen bond is formed and a carbon-carbon bond is broken. And as we can see, of course, the covalent bond intermediate is located below the reactant energy asymptote and shows a higher exothermicity than the van der Waals adduct. So the future perspective for this work is a complete analysis of the potential energy surface, considering all the possible isomerization and products, both considering the oxygen in the triplet P state and in the first electronically excited state singlet T, and considering also the possibility of intersystem crossing, which allows from the triplet surface to reach the singlet surface in which the intermediate are lower in energy. The data coming from the full analysis of the potential energy surface will then be used for a kinetic analysis in order to derive the rate constant and the branching ratios to compare with the cross-molecular beam experiments in order to derive the reaction mechanism. So in conclusion, we can say that we performed a benchmark analysis from the reaction of oxygen triplet P with acrylonitrile. As far as geometry optimization is concerned, all methods seems to provide similar results for what concern the covalent interaction. And the best candidate, the best compromise between chemical accuracy and computational cost appears to be the bit-relip functional, while for the long-range interaction, the best choice is the CAM-bit-relip, which is, as I said, an improvement of the bit-relip considering a correction for the long-range interaction. While for the energy, the best analysis that can be done is the coupled cluster, considering single and double, and the perturbative estimation of the triple excitation, considering the geometry optimized at the FT level. We noticed an agreement, a good agreement with the previous theoretical investigation. So the next step will be a complete PES investigation, considering the intersystem crossing, followed by a kinetic analysis and the derivation of the reaction mechanism by the comparison with the cross-molecular beam results. That's all. Thank you for your attention.
Thank you, Luca. Thank you for this nice talk. I don't know if there is some question online or from the persons that are in the room present. Some questions? Okay, I have a curiosity. So, when you finish to study this reaction, you will do the kinetic calculations.
In principle, yes.
And why in this type of reaction, so from astrochemistry, is interesting to evaluate the kinetic calculation, so the rate constant, which is
Because
the information that we can be obtained.
Sorry, I lost the last part.
Which is the information that we can be obtained in the kinetic calculations?
Okay, the kinetic calculations allow to obtain the rate constant as a function of temperature and the branching ratios. It's interesting to have a kinetic analysis because there are some astrochemical databases in which are listed all the possible reactions which can take place in the interstellar medium, and for these databases a rate constant is needed. So the rate constant is expressed as a function of different parameters which are inserted in these models.
Okay, perfect. Okay, thanks again, Luca.
Thank
And
you.
we move to the next speaker. That is Dr. Federico Palazzetti from University of Perugia. Federico, you are here with us online. So we wait. Can you hear us, Federico?
Maybe your microphone is off.
You cannot see. So we...
Ok, sì, eccomi. Posso sentire.
Okay, good morning, Federico. Federico Palazzetti from University.
Yes. Good morning.
He will talk about a minimal model of potential energy surface for the CO2-CO system.
Yes, just a moment. I'm trying to share my.
Yes, don't worry.
Okay. Can you see the presentation?
Yes, please. You
Okay.
can start.
Okay, thank you, Noelia. This work has been made in collaboration with Concetta Cagliotti, Noelia Pachinaslago, and Andrea Lombardi, and it concerns the construction of a potential energy surface for the CO2-CO system. That it's a system of interest in planetary atmosphere, because both these molecules are present in the Mars atmosphere and can be involved, for example, in the re-entry phase of space vehicles. We have realized an analytical model of potential energy surface. Here we present preliminary data. Analytical models are useful, are desirable for applications to molecular dynamic simulations and also for calculation of spectroscopic properties. Here we have realized a minimal model based on the expansion in spherical harmonics of the interaction between these two molecules. Both are assumed as rigid rotors. This approach consists in determining a minimum number of energy points by ab initio calculations. We have used in this case a Müller-Plesset of second order with a quadruple zeta basis set. These points are related to mutual orientation of the molecule that we call significant configurations. The spherical harmonics expansion represents an exact transformation of this quantum chemical input data. So it permits interpolation and possible implementation of sets of input data at a higher level of theory. Here I report the reference coordinate frame. The potential energy surface depends on 4 coordinates: the distance between the centers of mass of the molecules, 2 polar angles. Both are the angles between the molecular axis and the vector that joins the 2 centers of mass, theta A and theta B, and the phi, that is the dihedral angle between the 2 molecules. Here we report the significant configurations for which we have determined the energy points for a grid of radial distances, of radial points. So we have the parallel orientation, the linear orientations, depending on the proximity of oxygen and carbon, or oxygen and oxygen. a parallel, let's say a crossed orientation where the dihedral angle is 90 degrees, and also perpendicular orientations, and another orientation for which the theta angles are both 45 degrees and 45 and 135 degrees. Here, I report the expression of the potential energy surface that, as I said, is a function of 4 coordinates, is given as expansion of moments, expansion moments depending on the distance, and harmonics depending on the angular variables. In a more explicit way, the potential is expressed as a product of 2 harmonics, spherical harmonics, both representing 1 molecule. And here also we introduce the 3J symbol that expresses the coupling scheme of angular momentum. Here also report the explicit form of the potential of the interaction potential that is given by, as I said, the expansion of spherical harmonics and the related expansion moment. Here I report also the explicit formulation of the expansion moments. The first one, the 000, is represents the average of the interaction potential referred to obtained by averaging the significant configurations and represent the isotropic component of the potential. This is the isotropic component of the potential can be obtained also by experiments, cross-section experiment. Integral cross-section experiments. And so, if available, it permits to evaluate the quality of this potential of these calculations. The other terms are the anisotropic components of the potential, and all of them represent have a particular meaning in the interaction potential. Here I report the profiles of the significant configurations obtained by the energy as a function of the intermolecular distance. Here, for the first in the first panel, I report the H and the X configurations. Both present a minimum about 3.4 angstrom. The X configuration is more stable, as you can see. Regarding the collinear configurations, L1 is completely repulsive, while L2 presents a minimum, also Z1 and Z2. And for what concerns the perpendicular configurations, here we have the global minimum that is for T1, that is this T configurations with both carbons oriented one towards each other, and this is the same configuration found experimentally, and also the values are comparable. Here, the isotropic component that I remember is obtained by the averaging the intermolecular potential for the various significant configurations. And but in this preliminary data, these preliminary calculations, we didn't have performed the interpolation test that permits to evaluate the quality of the interpolation in those points that are not related to significant configurations, because here you can see that the expansion exactly interpolate the ab initio point. So I report another system, water HF, for which we have performed the interpolation test. In this case, the interaction potential depends also on an additional variable, angular variable, that is alpha. Alpha represents the azimuth angle of HF with respect to the water. So the significant configurations are much higher, because we have considered also this farther angle. So we have about 30 significant configurations, and okay, here I know I don't report the explicit form of the potential energy surface, but of course it's more complex than the previous one. But I report just the interpolation test. We have calculated so ab initio points and expanded the interaction potential for these three configurations that we called test 1, test 2, and test 3, for which I report also the values of the angles. In this case, the ab initio points have been calculated by a coupled cluster with complete basis set that is given by an extrapolation of MP2 with 3 basis sets, and corrected by adding the energy calculated by coupled cluster double zeta. As you can see, for 2 attractive configurations, there is a good agreement, much less than where the error is much lower than the computational accuracy of 1 kilocalorie. For the test 1, as you can see in the repulsive part, there is a certain discrepancy. Also, we have calculated the global minimum, and we compare the ab initio calculations with the 2 functionals, the Cambit relief and the LC omega PBE. As you can see, there is an overestimation of the well depth for what concerns the Cambit relief, and the subestimation for LC omega PBE that is more evident for the isotropic moment, where you can see the isotropic component calculated by LC omega PB is much lower in the minimum. In conclusion, we have proposed a minimal model for of the potential energy surface for 2 systems, CO2, CO, that we have seen, for which we have seen the expression potential energy surface, and water HF. These are, as I say, the preliminary data future works will aim at improving the accuracy by increasing the level of theory of initial calculations, and the applications of potential energy surface will concern elastic scattering simulations. And we presented a comparison of coupled cluster for water HF with 2 functionals, and we could see that for LC omega PB the potential is more repulsive in the short range distance with respect to the other functional and the coupled cluster, and the less attractive, especially for those configurations for which hydrogen bond is prevailing. On the other hand, Cambit relief is more attractive with respect to coupled cluster, and it tends to emphasize especially those interactions between fluorine and hydrogens. Thank you for your attention.
Thank you, Federico. Thank you. Thanks a lot. Very nice talk. Some questions, some suggestions from the people here or online. So, a curiosity, how do you think how to evolve this work in the future? So, do you think use this model that you developed to another systems or not?
Sorry, can you say the last part? I didn't understand.
So, can you say me about some perspectives about this model in the future for another systems or?
I think that we can, okay, first of all, we can improve this model, okay, by increasing the level of theory, and we can apply it in molecular dynamics simulation. For other systems, yes, we can use systems of the same degree of symmetry, or even by increasing the degree of symmetry by consider, for example, those molecules of interest in atmosphere, for example, water, CO2, or water CO. We can include oxygen. Yes, other than, okay. Is it okay?
It's okay. Thank you again, Federico.
Thank you, Nadia.
Bye-bye.
Bye.
We move to the next speaker. Is a speaker in presence, Dr. Lombardi from Peru University. He will talk about classification of biomolecules by invariant shape coordinates and the formation index. Please, Andrea.
Prof. Palazzetti, can you stop the screen sharing, please? Yes, thank you.
Okay, thank you.
Okay, good morning to everybody. My contribution is about large biomolecular structures, and I want to show you a systematic way to induce some kind of classification of such very complex structures. Okay. Let me give you a motivation for this kind of research. Of course, you know that biomolecules, for example, protein, are huge molecular systems. So you have hundreds, thousands, or tens of thousands of atoms, which are organized in a complex way in space. You know that there is a very complicated, complex relationship between the biofunction of these molecules and their structures. And one of the ways that we can use to classify this, the huge variety of structures, it is assigning to each structure a reduced set of parameters that in some way can take account for specific properties. Of course, this operation generates a classification in the sense that many structures are divided into subgroups. If you change the choice of the parameters, you change the view. So the trick is to find some proper way of defining a few parameters, a small set of parameters. Our idea here is to use global shape coordinates. These are essentially 2 or 3 parameters that are able to capture some of the properties of the global structure of these complex systems. We will apply quickly, as an example, these parameters to 2 opposite limiting cases. Clusters, which are huge, big systems, but with a very uniform distribution of the atom in space. And on the other side, proteins, which are big systems, but with a very detailed distribution and non-uniform distribution of the atoms in space. Let me show first how we can obtain in a systematic way shape coordinates. Starting from a complex structure. We have to manipulate the Cartesian coordinates of the system, and we can do that by performing first a scaling of the coordinates by masses. Then we take the components of the position vector of each atom and put them column-wise in a matrix. Once we have this matrix, this coordinate matrix, we can perform 2 kinds of operations. Actually, we realized that we can use orthogonal matrix to perform ordinary rotation in space, so a rotation of the global system, of the system as a whole. And also we can perform, using orthogonal matrices, kinematic rotation, which has rotation which take place in the kinematic space, not in actually physical space, in such a way that this kind of motion, the motion that are generated, tend to exchange the particle position. Why I'm doing that? Because we want that these 2 or 3 shape coordinates are invariant with respect to any possible kind of rotation, which are not only ordinary rotation, but also kinematic rotation. In practice, we isolate the only 3 parameters that are unchanged if we perform rotations and any kind of motion which locally do not modify the global shape of the system. This is the matrix theorem that we use to generate these parameters, which is the singular value decomposition theorem, which is a way to assign to each matrix, in this case our coordinate matrix, the product of 3, in a unique way, the product of 3 matrices. We can easily recognize here the R and K matrices, which correspond to the ordinary and kinematic rotation. There is this matrix in the middle here, which whose entries are all zeros, with the exception, with the possible exception of these 3 parameters, which are precisely the quantities that we were looking for, invariant with respect to any kind of rotation. There is a relationship of these quantities with the principal moments of inertia, and this is helpful because we can establish a relationship with the rotational symmetry of the system, of the global system. So we have conditions for a spherical top arrangement of oblate and prolate, and this fact suggests us that we can combine these parameters, these global shape coordinates, to generate deformation indexes and a quantity that it is called hyperradius, which is a measure of the real size space, in physical space of the system. So we have these 2 deformation indexes and the hyperradius. I have to go fast, so I'm going to show you some preliminary examples. First, the very simple case, trivial case of clusters. So let us have a look to Lennard-Jones clusters. So we can collect minimum energy structures up to 150 atoms, and we can simply plot the deformation indexes as a function of the size And what we observe is that the zero line contains spherical top structures, while points far from the zero line indicates that the structure is deformated with respect to the perfect spherical shape. Also, we can plot the hyperradius. We can see a not really linear, but a monotonic increasing trend with some outliers. I go fast, of course. Bigger systems, bigger clusters, same story for the trend with some outliers. And different kind of clusters, for example, C60 clusters with simulated by the Gini-Falco potential, minimum energy structure, and we can see a similar pattern for both the deformation indexes and the hyperradius. Here you can see that there are much more outliers, and this indicates that probably the search for the minimum energy structure should be performed in a better way. Metal cluster, just to show another case and to understand, I want to make you understand that the situation is the same, whatever kind of system we are analyzing concerning clusters. So this kind of analysis is not really suggestive, in my opinion, for a simple system in this way, but things are much more interesting for proteins. So we are talking about proteins. We have this huge structure. Of course, we can just assign a single triple of parameters to the global structure, but this is not the best approach. Probably the best approach is to follow the detailed structure of proteins as a sequence of amino acids. So we want to look at each individual amino acids, the shape of each individual amino acid, which is contained in a given protein structure. So in this way, we have, starting for even a single structure, a huge number of size parameters, and we can use them to make some kind of mapping, looking for patterns or regularities and whatever we notice. And so this is just the way we want to look at individual amino acids. We look at them as mostly tetrahedral structure, just because we know that these are chiral units, and the core of any chiral system, it is a four-center arrangement. So the alpha carbon of amino acid is in the middle, is at the center of a tetrahedral arrangement of atoms or centers or group of atoms. Then this is the backbone. We have a sequence of chiral units. For each of them, we can generate a triple of parameters, and we have to imagine, to guess a way to map those parameters. And so we can get inspired by the very well-known Ramachandran plots for proteins. So these are 2D scatter plots, actually, where the pair of dihedral angle of any single chiral unit in the backbone, of any single pair of chiral unit in backbone, it is characterized. So we have this kind of plot, and it is very, very typical for proteins. And the use of this kind of plots, it is, for example, to signal bad structures, bad 3D protein structures, because if the distribution of the angles is very different from the map which is generated historically, analyzing all the protein structures which are known so far, which are hundreds of thousands, this is an indication that probably there has been something wrong. So we can make similar treatment with these parameters. We can generate scatter plots from a sample of 500 NMR structure of proteins. We can scatter plot pairs of size. So we have these three possible scatter plots, the green, red, and blue, and we can see that actually there are well-defined regions for these parameters with some strange feature also. Look, this shape here and this line here. Okay, and we can merge together, of course, this the three scatter plots. So this should be an equivalent, something equivalent to the Ramachandran plots, but with a parameter more. And the use of such kind of mapping could actually be in the sense of making evident anomalies in the structure that are being measured or calculated for proteins. Another kind of approach for the use of these parameters, it is looking not at all the amino acids, but at just 1, 2, 3 single amino acid in the protein structures. We can, for example, consider 3 of the most common, most diffused amino acids in proteins, which are glycine, proline, and alanine, here in the figure. And just to start, we can consider 4 individual protein structures chosen randomly from the protein data bank, just to test this approach, and try to plot deformation indexes considering 4 proteins for these 3 amino acids. And we can see that a clear shape, it is visible here for the first case, protein case, similar shape for second protein, and similar for the 2 more proteins. This is a very small structure, so we have a reduced number of amino acids. That's why a few points. Of course, to check if it is something really significant, we have to use all the same protein sample, 500 proteins, and here is the results that we can obtain. This is the scatter plot of the deformation indexes for the entire set of proteins. And glycine, proline, alanine, it is a kind of fingerprint, as you can see, and the same story is more or less what we find for scatter plot of the individual shape coordinates. Okay. Well, we can also try to modify the way we calculate we identify the chiral units, obtaining slightly different results. I'm stopping here because I think I'm running out of time.
2 minutes.
Yes, okay. This is, for example, a different way of calculating the masses corresponding to the 4 units of the chiral, 4 center chiral unit. And so that's it, essentially. Let me draw conclusions. And so apparently these shape coordinates, which are invariant with respect to any possible kind of rotation and kind of motion that do not affect the global structure, can be used systematically to classify somehow by this kind of mappings large molecular structures, essentially proteins, also DNA, of course, with the same philosophy. And these kind of plots can be generated in many ways. I just was showing just a few of them. And so we can generate 2D plots of this kind. Next step, we can try to identify patterns, not only for the structure of the amino acid, but for the structure of sequences of amino acid corresponding with typical motifs of protein structure, like helices, for example, and beta sheets, and so on. Also, it could be interesting application to DNA. And third, chiral aspects. So we can use this kind of indexes, which are connected also to the chirality of these units, to generate a kind of measure of the degree of chirality of the amino acid in the backbone of the proteins. Thank you for your attention. I'm concluding here.
Thank you, Andrea. Okay, thank you, Andrea. Some question in the audience online. So, I'm curiosity. Sorry, you have a question? Please.
Can you approach for the microphone, please?
Sorry.
Can you approach for the microphone because people cannot hear you. Can you hear?
Ah, no, I can.
No, non sta funzionando quello.
Ah, no.
Qua.
Ah, sorry.
Please.
Okay, you tell me.
Here?
Here.
Yes, I think that the microphone è con la telecamera. I think it's in the camera. Please come.
Okay.
I think here is fine.
Okay. Okay, please.
You have these abstract models. I was wondering how do you describe like interactions such as hydrogen bonding, because you're aiming to go towards DNA. So I was thinking because it's similar to the coarse grain approach. So you have this abstraction of your atomistic level to a different model. How are you aiming for this atomistic interaction like hydrogen bonding?
Of course, this kind of approach can be also used to characterize the hydrogen bonding. Of course, interactions and potential energies here are not in the play, but we can just try, in principle, to look at the parameter distribution of subunits where we know that hydrogen bonding is a fundamental part of the interaction. Then we systematically can examine a huge number of structures, just plotting, making mappings for the subunits where we know that hydrogen bonding is, looking for some kind of pattern. It's just, it's not possible to know in advance what the results will be. But,
Yeah, okay, thank you.
Thank you very much, Andrea. Okay, we move to the next speaker. Okay, we move to the next speaker. Please, is there in the online audience Vadim Lisitsa?
Yeah, I'm here.
Hello.
Hi.
Hello.
Hello.
And the next speaker is Vadim Lisitsa from the Sobolov Institute of Mathematics from Novosibirsk in Russia. Sorry for the pronunciation. And he will talk about of a numerical simulation of the reactive transport at 4 scale in 3 dimensions. Please, Vadim.
Thank you for the introduction and good morning or good afternoon to everybody who is online. And so the my talk is not going to be about molecular dynamics, unfortunately, even though it was extremely interesting to listen to the previous talks. But my talk is about continuum, and what I'm gonna talk about the simulation of reactive transport. And typically, if people are talking about reactive transport, especially in the application to geosciences, it is usually assumed that we simulate the fluid flow using Darcy at Darcy scale. We simulate fluid flow in the entire reservoir to see what would happen if we do CO2 sequestration or enhanced oil recovery or something like that. However, if we are talking about reactive transport simulation at the reservoir scale, the standard models include such parameters like permeability and then efficient diffusion coefficient in porous media. but these guys actually depends on the porosity and on the geometry and topology of the pore space. And in standard models, the porosity is one of the variables in the model. But then permeability and then diffusion coefficient somehow depends on this parameter, and these dependencies are typically empirical. And then what we are trying to do in our work is to do simulation at the pore scale to construct the dependencies between the porosity, between the geometry of the minerals or of the rocks at the pore scale, and efficient parameters which are used at the reservoir scale. Actually, just pretty much the same problem appears if we're talking about seismic monitoring of I don't know, CO2 sequestration. When you inject the CO2, you typically so the carbonates may dissolve due to the CO2 injection, and then the mechanical properties will also change. And these changes strongly depend on what is going on at the pore scale. Actually, same as permeability and diffusion. So what we are doing, we are considering the fluid flow at the pore scale. We know the geometry of the, we know the model of the pores of porous material of porous carbonate rock, which these models typically come from CT scans at the macro scale, at the micro scale, and we simulate the fluid flow in there and the transport of the reactant. So in this particular system, well, this particular equation governs just propagation of one reactant, but of course it can be much more complex, and quite a lot of species can be there. And then what is the most important thing here, that we are focused on the heterogeneous reactions, and we try to follow to simulate the evolution of the pore space, the evolution of the interface between the pores and the material and the core and the matrix of the rock. And this boundary movement is governed by quite a simple equation, which of course the velocity of these changes depends on the concentration and reaction rate and stuff like this. So all this come to this equation, and in this particular case, it was just a linear reaction, first order reaction. We assumed it, but of course it can be more complicated law in this equation. So, and this is actually the most difficult thing, the most difficult part in the simulation. Of course, like solving Stokes equation, simulating the solving advection diffusion reaction equation in the pore space is not that easy thing to do. But the most difficult thing is to simulate the movement of the interface. And to do so, we are using a level set method, which allows us not to follow the interface exactly, but to define the interface as a zero level of some artificial function. And we reformulate, and the equation of the interface movement can be reformulated as the changes in this artificial function. And then we can do it using just the same meshes that used for the simulation of the other processes in here. And the best thing about this level set method is that it easily deals with changes in the topology. So if we had a isolated pore somewhere, and then the matrix starts dissolving, and then the pore opens, and then join the connected pore space, it can be easily dealt using a level set method, apart from, for instance, front tracking methods. So, and that's pretty much it about the theory and the math. And if you wanna, you may ask me what numerical schemes I use particularly, but okay, let me proceed to the numerical experiment. And first series of experiments was done for the to validate our approach against the laboratory experiment. And you can see the reference over here, the quite a nice paper on laboratory experiments where the species of calcite and dolomite were used, and CO2 was injected in there, and the rock was partially dissolved. And the authors managed to get the CT scans during the dissolution process, and they had they did it for 4 different samples, and they had 10 snapshots of the process for each sample, and we tried to reproduce it. So we took CT scans corresponding to the beginning of the process, at the very first stage of the process, and then simulated CO2 injection and dissolution of the matrix, and see if we are close to what was done in lab. So apparently it appears to well, apparently we had a quite a nice correspondence in the changes of the porosity and permeability of the sample with quite a nice so the correspondence between our numerical simulations and the lab experiment was quite nice. So when we went further, and we decided to see, of course, changes of the porosity itself is nice, but it's not the probably not the main parameter which governs the permeability of the sample. And if we are talking about CO2 sequestration or chemical reactive transport, it quite oftenly it leads to the wormhole formations forming when the permeability gets anisotropic, and quite a small changes in the porosity leads to quite a strong changes in the permeability. And what we did, we tried to see what would happen if we play with the parameters. I mean, if we change the diffusion coefficient, molecular diffusion coefficient in our equation, if we change the reaction rate, if we change the flow rate, and what would happen then if we can classify somehow the difference in the of the changes in the pore space. And to do so, we apply the we appeal to the topology, and not just topology like classification of how many connected bodies or cycles we've got in our pore space, but there is an approach called persistent diagrams, which allows us to see how the topology evolve in time. And that's quite a nice thing to do, because we can actually distinguish between completely different scenarios of the matrix dissolution. And here's an example. Actually, there are 4 examples. We started with the same model. I mean, the same model for the pore space, and we started simulation using completely different like reaction rates and flow rates and so on. And then we estimated the changes in the topology and classify different scenarios according to the topology changes. So on the right, you can see the persistent diagrams, which shows how many cycles in your pore space, and when they appear and then they disappear. So the persistent diagram illustrates the moment when the particular cycle, particular channel appeared in the pore space, and then and when it disappears or dissolve due to the dissolution of the matrix. And you see that we can use this information just to classify different scenarios, and you see that the changes in the pore space are completely different if we
Balin, you have
Yeah.
2 minutes.
Yeah, great. So, and then we see if we have a correlation between these topology changes and the input parameters which we were using, and indeed there is quite a huge, quite a nice correlation between them. And if we classify our experiment according to the topology changes, you can see how they are how well the agreement with the changes in the physical properties of the experiment. And then of course we plot the dependence of porosity and permeability on time for different clusters which were clustered by the topology changes. And then we plot the cross plot the porosity permeability to see that according to different changes in the topology of the pore space provides us with completely different cross relation between the porosity and permeability, and we need to take it into account when we substitute such relations to the macro scale model at the Darcy scale. So that's pretty much it. Thank you for your attention.
Thanks a lot, Vadim. Is there some question in the audience? Online, for the persons that are online? Okay, so just a curiosity. I can read that the algorithm is implemented on GPU. I would like to know if you compare the computational cost with other clusters or machines, or you implemented it directly in GPU.
So this particular algorithm was implemented directly on GPU because when I tried it for the first time, it was 2D simulations, and I compared it for 2D simulation. I compared it with the CPU implementation. But when when 3D algorithm was implemented directly on GPU, and we had no comparison with CPU simulation, unfortunately. Or fortunately, I don't know, because we would never have enough time to wait for the CPU to compute.
Okay. Thank you again, Vadim. And we move to the next talk about Miss 4 minutes. We wait 4 minutes and or split this talk.
I'm sorry. May I try to share my screen so that I check if the presentation works, so that I see if my slideshow works.
Streaming is on.
Good morning, Noelia. I would like to check if my slideshow works in this GT software.
Okay, but I think so that we are late.
Okay, all right then.
So you are the second speaker after Damiano when arrive.
Mm-hmm.
And don't worry.
All right.
Don't worry. After that, we prepare.
Give us 1 minute, please.
We try to introduce the Damiano.
No, not yet. Just give us 1 minute, please. We are a bit late.
Yes, thank you.
Okay.
Okay.
Okay, Okay, hello. So we start with the second session of this morning, and I would like to introduce Damiano Perri from
The University of Florence.
And he will talk about a new method for binary classification of proteins with using machine learning. Please, you have 12 minutes.
Okay.
And
Good
3
morning.
minutes for the questions.
Yes. Okay. Perfect. Good morning to all. I'm a PhD student at the University of Florence, and I am part of Professor Osvaldo Gervasi research group. And I'm here to present our latest work. We have developed an approach based on artificial intelligence algorithms that allows us to classify true proteins from false proteins that do not exist in nature. And in this presentation, we describe a deep learning based approach. And we use a convolutional neural network. Oh, sorry, the screen goes down. Okay. Okay, perfect. And as I said before, we used a convolutional neural network to categorize protein chains of amino acids based on the protein description contained in the protein data bank, the PDB database. Each protein is fully described in its chemical, physical, and geometric properties in a file, an XML file. The XML files describing the proteins are all available free of charge on the web by accessing the protein data bank, which has been crucial to our work. In addition, XML files are easily analyzed by modern programming languages and allow you to quickly extract all the information you need for your research. And we wanted to apply the proposed techniques to other general classification issues in biomolecules, such as structural similarities and differences. And okay, we can go on. The convolutional neural network was chosen as the network to handle the sequences. And moreover, we used some techniques, some advanced techniques, for example, the transfer learning. We start from a pre-trained network, for example, VGG16 or Inception ResNet and so on. And we freezed all the layers of the network except the final layers that we used those layers for training the learning to learn how to classify the proteins, because the first part of the network extract the futures of the input, and the last layers will classify the futures and perform a binary classification, because we just need to say if a protein is true or false. Okay, thanks to this technique, we were able to speed up the learning and test more neural network model. Okay. Our work was fully created in Python 3, and we used the Google Colab platform as developing environment. And we used some CNN, for example, is Inception ResNet v2 or Inception v3. In this picture, we can see how we start our work. For example, we got all the data set from the protein data bank, composed of hundreds of thousands of proteins, entirely described by single files, and we read all the information that we need. And the next part was the data extraction and the processing of the information contained in the XML files. We collect 3,000 instances, 3,000 molecules, with a total of 16 millions of amino acids. And after that, we applied data augmentation. Our data augmentation consists in a data set with the protein read from top to the bottom, and the same proteins, but read from the bottom to the top. In fact, we doubled the number of elements in the data set. After that, in the second phase, we associated a unique RGB color code to each amino acids in the proteins, in order to visualize the structure of the molecules as an image, on which every single amino acid is colored in a different way. Okay, the most important part of the presentation is that we used a orthographic axonometric map. We use orthogonal projections on the three planes of the molecules. But why we do that? Let me show you an example of how we compose the images of the proteins. Here we can see a geometrical schema
that we used for the picture, for the creation of the pictures. And here we can see an example of how a protein is represented on our images. All the images have a fixed width and height, and here we can see the most important slides. This representation is the heart of whole our work. Molecules have 3 dimensions: the X, the Y, and the Z, which represent the spatial coordinates of each amino acid. In addition to this, each amino acid also has a name, and the name of the amino acid can be seen as a 4th dimension. In other words, our problem is a binary classification of a 4-dimensional problem. And thanks to this representation, we have reduced the size of the dimensions from 4 to 3, as the 3 views represent the molecule exactly, and there have never been any overlapping amino acids based on our tests. It is possible to take photographs of the same molecule seen from different angles of rotation. This approach was crucial as convolutional neural networks require 3-dimensional images, the width, the height, and the color. And we were therefore able to use networks that obtain 3-dimensional input to work with a 4-dimensional problem. And okay, to produce the fake samples, after that, which are required for neural network learning, it was chosen to begin with the original picture, and we apply some mutation using a mutation probability of the amino acids. And with these techniques, we generate the fake protein used for the classification. In this image, we can see a real protein at the top, and at the bottom, a false protein. The deconstruction phase of the neural networks was described in this part. We use pre-trained the network and trained just the last part of the CNN. Here we can see the models that we use and the descriptions of the models. And here we can see some result. For example, the accuracy of the protein recognition on the train set, and the accuracy on the test set, and the model size. Okay. Finally, let me show you some confusion matrices, where we can see that the model performs very well on this classification protein problem. For example, we can see in this confusion matrices that the inception view3 perform very well. Okay, in conclusions, some considerations about the generic nature of the CNNs employed led us to believe that particularly constructed neural networks might considerably enhance, if not outperform, the results. And a second research topic worth investigating the training of the neural network with a larger number of samples in order to better understand the relationship between the structural complexity of the samples and the neural network classification capabilities. Okay,
Thank you.
Thank you, Damiano. Nice, interesting talk. There is some question online here. Okay, thank you again. Okay, Andrea, please.
Do you think that this kind of approach can be also applied to other similar molecular systems,
Yes.
such as DNA, and just to identify anomalies and
Yes, I think that we can use an approach like the one that I described before for some other problems. And yes, I think that can be used for DNA-related problems, of course, yes.
And my question is, if you just substitute your XLM files
Yes.
For proteins with XLM files for DNA structures, works as well?
Maybe we just adjust some piece of codes, but I think that with some short tuning, fine tuning of the code, yes, I think that it should work for classification problems. Yes.
Okay.
Thank you again. Thank you, Damiano. We move to the next speaker.
Okay. Bye bye all. Thank you.
Okay, so Yogan, can you hear me?
Yes, yes, very well. Thank you very much, Noelia.
I see your share presentation. Okay, I would like to introduce Yogan Tolbatov from the Institut de Chimie at the Université de Bourgogne in France. Please, Yogan will talk about multi-layered modeling of the metallation of biological targets. Please, Yogan.
Thank you very much, Noelia. Now the first slide of my presentation can be seen. Yes? Is it correct?
Yes.
Yes. Okay. Thank you very much. So good morning to everybody. Thank you very much for giving me the opportunity to present here the various computational strategies for the multi-layered modeling of the metallation of biological targets. As you know, metals in biological systems are not that easy to be described. There are several reasons for that. First of all, the metal cations are very promiscuous. They tend to interact with a plethora of various biomolecular targets. Secondly, metals possess an exceptional adaptability of coordinative bond to their ligands due to the reciprocal adjustment of the electronic structure of both the metal center and the ligands. And the third characteristic feature here is the substitution mechanism of an attack by a metallo drug. So when a metallo drug, in order for the to perform an attack on its target, metallo drug should first lose the coordinated ligand, and afterwards this vacant position should be occupied by the biomolecular target to which the metallo drug is attached. So when one speaks about the metallation of biological targets, there are two major directions here. These are metalloproteins and metallo drugs. So 50% of all proteins in the human body are based on the metals. They are metalloenzymes or metalloproteins. So they are of all six classes, as you can see. The usual metals in the active sites of these metalloproteins are zinc, magnesium, and iron. In order to study the functionality of these metalloproteins, it is necessary computationally. It is necessary to use some special approaches because metalloproteins are very, very big. And that's what we are going to talk about now. And the other direction here is the metallo drugs. So as you know, bacteria always evolve evolutionary, and they constantly evolve new defenses against the antibiotics. And a new fashion here, new trend, is the synthesis of metallo drugs, which target the bacteria or the viruses. So this is another direction about which we are going to talk today. So the metallation of biological targets, always always we talk about something very big. It is always a protein, it is DNA. There are several thousands of atoms. It's impossible to study such a big system with a single approach. That's why the separation of studied system into zones is crucial here. One can separate always a studied system into a zone, smaller zone, that is treated with an increased accuracy, and the greater region, the effect of which is taken into account with the employment of a better scalable methodology, which is less precise. Usually it is the empirical force field. So one of these separation techniques is the separation of the studied system into the bulk layer and into the target layer. So usually this approach we use when we study the metallo drugs. So the bulk layer is the layer where the activation of the metallo drug occurs, whereas the target layer is the layer where its interaction with the biomolecular targets takes place. Here I will show to you several examples. So, first of all, cisplatin. As you know, the serendipitous finding of this metallo drug served as a crucial step for the development of modern bio-inorganic chemistry. Cisplatin, you see, it is activated by the hydrolysis. So one of chlorides is substituted with water. So this process happens in the bulk layer. Afterwards, when this metallo drug is activated, it interacts with its target. Its target is DNA. Usually it is guanine, although there are some intrastrand and single-strand interactions. But generally, interactions of this metallo scaffold with its targets happens in the target layer. Another example here is we have studied various derivatives of cisplatin. One of these derivatives is bispidine analog of cisplatin. You can see here bispidine is an anti-tumor drug that can be attached instead of two cis amino groups at cisplatin scaffold. And this metallo drug can interact with the guanine. However, it should be firstly activated, and its activation happens in the bulk layer. By means of hydrolysis, either one or two chloride anions are displaced and replaced by the water molecules. Afterwards, the activated bispidine analog of cisplatin interacts with the guanine, and as you can see, the activation energies of this metallo drug are comparable to the activation energies of cisplatin. The same approach was utilized for the studies of carboplatin. Again, activation of this metallo drug by chloride anions was studied in the so-called bulk layer of the simulation, whereas the target layer analyzed the interaction of this activated drug with its biotarget with guanine. By the way, what was found here is that in the weak acidic conditions, this melanate ring that is attached to the platinum center turns out to be quite stable, and a high activation barrier, 27.5 calories per mole, tells us that this metallo drug is not activated in the weak acidic conditions, whereas in the strong acidic conditions it is more labile. The melanate ring, this bidentate ligand, is more labile, and it loses the platinum scaffold loses this ring quite easily, and the activation energy for the first, second activation steps quite low, around 15 kilocalories per mole. So all of these results are obtained with this logic. So the activation of the Metallo drug may happen also electrochemically. For example, one of the strategies to circumvent the toxic side effects of cisplatin is to use platinum-4. Platinum-4 scaffold with 6 ligands, which are afterwards activated electrochemically, and cisplatin is produced. So very often the XL ligands are added onto platinum-4. Usually these ligands bear some anti-tumor organic molecules. And experimentally, it was impossible to study this two-electron reduction process. However, computationally, we were able to disentangle this problem. We have found out that firstly, platinum-4 is reduced to platinum-3. Afterwards, the first XL ligand is lost. Afterwards, platinum-3 with 5 ligands goes through a concerted step when platinum-3 is reduced to platinum-2 and the XL ligand is lost, and so cisplatin is formed, and this cisplatin goes to interact with its biomolecular target. Again, there is a bulk layer in which the electrochemical activation happens, and the target layer in which already the interaction of the Metallo drug with its target takes place. Another logic which can be applied here is the usage of two layers, layer and QM layer. This approach is usually used when we study the proteins. Proteins are very big, thousands of atoms. So whole protein is studied with a less accurate method based on classical mechanics, empirical force field. And the site of the protein where the chemical reaction that we are especially interested in is characterized by quantum chemical approach. This thinking allows us to analyze big systems. For example, recently we have studied this type of Metallo drugs based on ruthenium, osmium, rhodium, iridium, and N-heterocyclic carbines. So their interaction with the hen egg white lysozyme, it's a typical protein. Usually it is used for these studies. So we have used here 3 levels. The bond dissociation energies of various ligands were studied with quantum mechanical level, so it was very accurate. And we have found out that the most labile ligand always is chloride. But in the case of ruthenium, thymine, surprisingly, which is eta 6 coordinated, also can be quite easily lost in case of ruthenium. QMMM level was used for studying these Metallo drugs when they are attached to the protein, so that we were able to take into account the effects of the biological milieu around this place, and the spherical effects maybe of the protein, whereas the dynamics of the whole protein was studied by means of the approach with a classical force field. The same type of thinking was applied when we studied metal binding proteins. For example, metal binding protein that is very selective towards gold plus cations. That's a unique protein which is used when gold is lost in the environment, and already there are the examples when this bacteria, Cupriavidus metallidurans, with this protein was applied for the detoxification of the environment. So again, look, level was used for the studying of the whole protein, whereas the metal binding motif based on 2 cysteines, which can be neutral, deprotonated, this exactly metal binding motif was studied by means of quantum chemistry. And we have found out that this protein possesses the unique capability of either have the, it is a molecular switch. It can either chelate gold plus cations, or it can make them free depending on the pH of the biological milieu in which it is situated. And the last example is the metal binding protein PBRR, which has a specific selectivity towards lead. Again, it is very in this industrially perspective because it is a unique natural protein with a special selectivity towards Pb2 plus cations. Again, level allowed us to look at the whole protein, which possesses 2 metal binding sites, whereas the metal binding sites, which are composed of 3 cysteines, each of which may be neutral, deprotonated. Also, there are some loops around these cysteines. So this exactly fragment was studied by means of the quantum chemical approach. So, in short, I have described to you the strategies that are utilized for the studies of the metallation of biological targets. This problem is quite a difficult one because of the presence of a bunch of valence electrons in metals, and usually you cannot, when you study this in a quantum chemical way, you should always take into account the relativistic effects. Usually the effects of the inner layers of electrons are taken into account by the effective core potential. So these systems are not that easy to be studied. And in order to make it possible, we need to utilize not a single approach, but an approach when the studied system is separated into 2 layers, and there may be different logic behind this separation. However, this separation is of utmost importance for this type of problems. I would like to acknowledge the supervision of my current supervisor at the University of Burgundy, Professor Paul Fleurat-Lessard. And all this research that I have presented to you was done in collaboration with my former supervisors and current colleagues from the University of Chieti Pescara in Abruzzo, Professor Re, Professor Caletti, Professor Marrone, and also with collaboration from another postdoc, Dr. Roberto Pacciotti. Thank you very much for your interest, and I am eager to answer your questions.
Thank you, Jogan, for this nice, interesting seminar. Some question in the room or online. There is the people online that maybe wanted to do some question. Any questions? So I have just a curiosity. So the next step in your case, what do you think the another biological system to study? You have a line of this, a lot of pieces that you want to study, or there is a line from smallest to more bigger system?
I think you are asking to which extent how scalable this approach can be. I guess this is what you are asking. I can tell you, the first separation, the separation into the target layer and into the bulk layer, is better applicable for the studying of the Metallo drugs. Metallo drug and, for example, various protein side chains. The second approach that I described is better described for the proteins which include the metal proteins. However, it is very difficult to parameterize the metal ions for these classical force fields. So the second step is, I am sharing the screen right now. Look, it is the slides that I have skipped. So the next step here is, look, we can use the cationic dummy models of the metals. Do you see the screen now? Yes? Is it correct?
Yes, yes.
Yes. So, for example, what we have found out that in case of some proteins, the metal cations, when we include the metal cations into the simulation, they just escape into the bulk water. They do not want to be coordinated in the protein. And we have found out a method to circumvent this problem. It is the usage of cationic dummy models, when a part of the mass of the metal cation and the part of the charge is distributed in some small number of dummy atoms which are situated around the central metal cation. For example, here lead with 3 dummy atoms, lead with 4 dummy atoms.
Okay.
So we study various clusters, and by this we take an insight into the geometry that these dummy models should preserve. And afterwards, this simulation, for example, we have done it already on the lead binding protein. We have found out that by usage of this model, we were able to obtain the coordination of the metal cation in the classical force field, in the binding locus. So it means that this approach may be used for the bigger systems, so for the metallo enzymes interacting with metals, but we may gain large trajectories by using these cationic dummy models, which allow us to play with the electrostatic and the van der Waals interaction of these metal cations in the biological milieu. So that's another step in the scalability of this logic that I have described today to you.
Okay, thank you very much for the explanation. Okay, thanks again. And we move to the next speaker. Emilia, are you there?
Yes, I'm here.
Okay, so I introduce Emilia D'Aragao, is from Peru University, and she will talk about long-range complex in the HC3M plus CN potential energy surface, using ab initio calculations and intermolecular potentials. Please, Emilia, okay, we will see your slide.
Yes.
Please, thank you.
Do you see everything? Okay, so thank you. So as a professor has introduced, I'm Emilia D'Aragao, and today I'm going to present my contribution to ICCSA. The title is Long-Range Complex in the cyanoacetylene and cyanoradical potential energy surface, ab initio calculations and intermolecular potential. I'll start this presentation by talking about the context I'm working with. So I am working with systems of reactions in the gas phase in the interstellar medium. Up to date, about 200 molecular species have been detected in the interstellar medium, and they are found either in the gas phase or on grain surfaces. So in the astrochemistry field, we are interested in understanding how these detected molecules are first formed and then destroyed. And the ultimate goal of this field is to understand how the molecular complexity of the universe has evolved over time. So in order to organize all the information about the mechanism of formation of molecules and destruction of molecules, we have to make astrochemical models. So astrochemical models combine observational data, by which I mean what kind of molecules have been detected, like it was shown in the table before, and how much of these species have been detected. We have also experimental data, where in the laboratory the experimentalists try to simulate the environment in which these reactions occur, and so they can derive information about the product branching ratio and eventually rate coefficients of formation. And we have as well the theoretical data, where we not only see what products can be formed in a reaction, but also what are the more probable products being formed, so the reactive channels, and so product branching ratios and the rate coefficients of formation. As a computational chemist, in order to build this model, well, in general, in order to build this model, we need to characterize correctly the reactions that can occur. And as a theoretical chemist, I started the work, the theoretical work, by drawing the potential energy surface. That means find each stationary point, it means the intermediates, transition state, and products. So let's say, so the rate coefficients are very important for the astrochemical models to explain the abundance of the molecules. And in the model that we usually use, we use this modified Arrhenius equation is expressed in cubic centimeters per second. So now into the reaction I'm interested in is the reaction between cyanoacetylene and cyanoradical occurring in the gas phase. Cyanoacetylene is a member of the family of cyanopolyenes and is formed at a very low temperature in the interstellar medium. It was detected also in the atmosphere of Titan and in the interstellar medium in dense interstellar clouds, as an example, Sagittarius B2. So it is known that this reaction with a cyanoradical gives only one isothermic product, that is the cyanoacetylene and the hydrogen radical. However, it is usually reported that the formation of this product occurs by direct formation of this minimum, the intermediate mean one that I've calculated as well, and then a transition state and formation of this product. However, when we perform the computations of the kinetic rate constant, we observe that we have a rate coefficient at 10 to the minus 10 cubic centimeters per second. This is the order of magnitude. And it is known in the literature and for other reactions that neutron-neutron reactions usually have an order of magnitude of 10 to the minus 11 cubic centimeters per second. So something is not being considered here, and that is making our reaction occurs much faster than it's usually observed for neutron-neutron reactions. We have to consider the back. In fact, we have to consider one thing: the back dissociation happening. Back dissociation occurs mainly when we have a van der Waals adduct forming, and then when this van der Waals adduct form, the energy is very close to the reactants, and so it can easily back dissociate into the reactants. The consequence is that the product is not always formed, and so we have a lower rate coefficient. So in this paper, we try to optimize a van der Waals adduct connecting the reactants to the intermediate mean 2 via ab initio calculations employing BT-RELEAP for the optimization and frequency analysis, and then we compute single point energies at a couple cluster level. So after many tries, we have managed to obtain this linear van der Waals adduct and this saddle point that connects into this bounded intermediate mean 2. However, if we look at the energetic profile, we can see that the van der Waals adduct is 10 kilojoules per mole below the energy of the reactants. And usually the expected value for a van der Waals adduct where back dissociation can happen is around 5 kilojoules per mole or less. So when we computed the rate coefficient for considering these 2 points, we again obtain a rate coefficient of 10 to the minus 10 cubic centimeters per second. So we thought that we needed to consider another van der Waals adduct, but after many attempts with the ab initio calculations, we did not find an adduct, a convergence of the structure that corresponds to this carbon-carbon formation. So for this, we decided to make a study using the semi-empirical methods to describe the approach between the two fragments. We consider the semi-empirical potential has a side contribution of non-electrostatic potential and an electrostatic contribution. For the non-electrostatic potential, we use the improved Lennard-Jones model using these parameters, and for the electrostatic part, we use a per-Coulombic potential with these parameters and charges. So as a result, for this linear approach of the van der Waals adduct that we calculated before, we have this dashed curve that corresponds to this structure being optimized at the couple cluster level. First, we perform this ab initio again, but we do not fully optimize the structure. We freeze this carbon-carbon distance and then compute the energies at optimizing all the bounds and the angles. But we freeze and obtain the couple cluster energy and obtain this curve at a different distance. We can see here that this is the region around which we found the optimized linear adduct. However, when we look at a very large distance, we see that the couple cluster method is not reaching zero as it would be expected. So this way, we performing the calculations with the empirical potential, also freezing this set of distance and approaching the fragments. We obtain this blue curve, and near at a very large distance, we have this potential close to zero. And so we thought that this describes better this approach of the van der Waals interaction. In what concerns the van der Waals adduct, we see here that the bottom of the well is more towards 5 angstroms, while here it's at 4.5 angstroms. So we tried to see other approaches, and we stumbled upon this orthogonal approach with this distance also being corresponded here. And first, we performed the semi-empirical calculations and found that we have a well around this region of 3.0 angstroms. So we tried to optimize the adduct around this region. Unfortunately, we still didn't have at the ab initio level the van der Waals adduct. But by performing the variational calculations where we freeze these points, we have found that there is a behavior of a well here, then a sudden augmentation energy corresponding to a transition state. We also tried to optimize this transition state, but it was not found also at the point of the publication. But so we have managed to characterize with the semi-empirical potential this van der Waals adduct, and it helps us to see another possibility of approach between fragments. And so the next steps are to see to perform a calculation considering this kind of adduct and see if it is if we get an order of magnitude that corresponds to radical-radical reactions. So in conclusion, a van der Waals adduct has been detected in the entrance channel leading to the formation of MIN2, the linear van der Waals adduct, but this approach of fragments do not explain totally the formation of MIN2. Then by applying the semi-empirical method, we detected another adduct at when the angle is orthogonal between fragments. Then we detected a barrier between this orthogonal van der Waals adduct and the intermediate MIN2. The semi-empirical method can help us characterize a region of the potential energy surface where we could not directly calculate with ab initio calculations because the calculation did not converge. And we also have a good, better description of a long-range interaction. So I would like to thank all the contributors to this work, and I thank you for your attention.
Emilia, for this interesting talk. Some questions in the classroom, in the audience online, seems so. Marzio, please. Okay.
I would like to know if you think about the possible different applications of this approach. I mean, the semi-empirical for long-range interactions and ab initio for the interacting part of the surface.
Yes. Well, mainly for trying to localize a van der Waals adduct that has not been localized, but also for understanding the longer range interactions.
Okay, thank you again. Thank you, Emilia.
Thank you.
Ok, rimango io qua, sì, mi autopreso, non c'è problema. Ecco, ce l'hai, a posto, me la metto in presentazione e mi metto seduto, così. Ah, ecco, Yes, downstairs. Con la freccia, no?
Così ma non vediamo cosa succede qua. Mettiamo proprio il PDF.
Ah, vabbè, ma uguale, no? O no? Come?
Just give us a second, please.
Okay, please, the people that is online, can you see the slides?
Yes.
Okay.
No?
Thank you very much. Qui non si vede, ma...
Ok.
Ok, adesso perfetto. Okay, I introduce myself. I am Noelia Fagina-Slago. I'm from Chemistry, Biology and Biotechnology Department from University of Perugia, and I will talk about of intramolecular force for the interaction of water, graphite in membrane contribution. So we analyze the contribution of induction effects. So this is the summary of these talks. Why we have decided to analyze the contribution effects of the water graphite system? Because in the last few years, we have studied the selective and capture gas, in particular of the CO2, obviously for the environmental situations. In the last years, we have involved in the study of gamma graphitenes using molecular dynamics tools. Specifically, we have created the new force fields to relate the gas absorption. Obviously, we have analyzed the CO2 for the small molecules of gases, like in this case, the CO2. And this is a graph that represents why it's important to study the the problems created from the CO2. Okay, so far we have analyzed that is very important, the study of the fraction of the total CO2 emission, because is necessary for the post-combustion carbon capture. We have analyzed the porous solid absorbance respect of the aqueous absorbance. At this point, a number of force field has been developed for evaluating the absorption of gas molecules for different porous materials. However, a challenge was made when in the mixture of the gases contain water molecules. So the idea is design and simulate novel porous absorbent predictions. So if in our system contained water molecules, this phenomenon indeed change in the intermolecular forces description, because the water molecules can modify the polarizability of the other water molecules near of the molecules in a study. Okay, this is the importance of water-water intermolecular intermolecular interactions. So far, we have treated the intermolecular interactions like a van der Waals plus electrostatic potentials using generic force field, UV, amber, but at this point we have neglected other effects like the induction. So in this work, we have analyzed the molecular dynamics simulations, introducing the induction effect. So we know that the intermolecular force of interest in our works can be defined like split in a non-electrostatic plus an electrostatic contribution. The non-electrostatic contribution is measured by taking into consideration the strength of induced dipoles and the average molecular size. So this, from the non-electrostatic contribution, this can be done by assigning a value of polarizability to the both interacting centers. In the case of electrostatic contribution, we have analyzed employing the Coulomb's law. So, in the case of the non-inductive part, so inside of the non-electrostatic contribution, we have adopted the improved Lennard-Jones potential. Is given by this equation where we can see a positive term that is the repulsion term and a negative term that is the long-range attraction term. We can observe also that in this formulation we have the epsilon is the well depth of the well depth. M can be adopted three types of values. 6 in the case of the neutral-neutral interaction, 4 in the neutral ion interaction, and 1 in the case of ion-ion particle. Okay, the important parameter in this formulation that is very similar to the Lennard-Jones, to the classical Lennard-Jones, is given by this parameter, the parameter beta. This parameter is a factor that modulates the hardness of the interacting pair. So this parameter introduced in the improved Lennard-Jones potential take account some effects of atom clustering, like induction and charge transfer. This type of potential was developed in our group at the university. When we speak about the charge transfer and induction effects, maybe important in the case of the water molecules, is important quantify this contribution. We have quantified this contribution introducing this part, the interaction of the induction. The induction is given by this formula, where nu is the dipole moment and alpha is the polarizability of water. This is the mu is expressed in the bar, in the alpha is expressed in angstrom cube. Uppercase R is the distance between the center of the mass for each water pair given in angstrom. C, the uppercase C is a constant equal to 316 to convert the potential in milli-electron volts. Okay, so now we move to our system. Here in this system represent the structural details of the molecular models in the case of the molecules of water, and in this case of the graphite membrane. The graphite is a form of graphenes where each of its benzene rings is connected to each of 6 others through a chain composed by 3 acetylene bonds. Here we represent the data of the structure of the water, considering that the water has a moment, a dipole moment of a 2.1 device. We run in the molecular simulations where we define the box filled with a random number of molecules of water in the both parts of the membrane, where the graphite layer is located in the middle of the box. Okay, here I will present the parameters for the non-electrostatic interactions. So remember, for these equations, for the water-water interaction and for the water carbon of the graphite membrane, okay, that we have used to define the force field in our system. Okay. At this point, we will like study the computational cost, introducing the induction contribution. At this point, we need to modify. We have modified some parts in the code. We have used the DL poly package. The choice of this package is how we can see in this case the possibility of write and modify the routines. So our modifications have been included in the following parts. We have modified the forces modules routine, and we have two pieces of routines inside of the Ewald modules and the Coulomb modules. So we can say that the Ewald modules call the improved Lennard-Jones potential, the Ewald modify call the improved Lennard-Jones potential plus induction effect, and we add a induct part that call the subroutine that modify together improved Lennard-Jones and induction. Okay, this is very important because This we have bright three Fourier cycles inspired by the counterpoise method. These three Fourier cycles is that are calling from inside this routine and to deal with the charge distribution flexibility of water molecules. What means it? That depends on the Fourier cycles that you choose, you can interchange the water atomic charges. So the total dipole moment can be moved from 1.85 device until 2.1 device. Okay. So this part is the part that from computational cost is the most time consuming part. Okay, here we have presented some CPU time for the simulation of pure CO2 with graphene using 600 femtoseconds. Obviously, you know that this time 600 femtoseconds is a not large time for a molecular dynamics simulation, but we was tested these new subroutines. You can see that the time is increased a lot from more or less 50 to 200, more or less 50. So in this case, we analyze how we can modify these routines to avoid this big increases. Okay, so in this table, I will show you the time and the results of the configuration energy, van der Waals and Coulombic energy using only improved Lennard-Jones and improved Lennard-Jones plus induction. Obviously, we can see that with dynamics of the 2.2 picoseconds, the times are more or less the same, but it is not enough time to consider that our system is converged and we can analyze the results. So for this motive, we observe what happened at 2000 picoseconds, and we can observe that the times is 4 times longer than the same system with the improved Lennard-Jones potential. So from the first results in the last year, we have improved the CPU time from the previous version codes. Obviously, we need to accelerate this time because we have analyzed that it's very important the introduction of the induction time to the contribution of the different types of energy. So here is from a chemical point of view, we can say that in this case, in the left part, we can analyze the density profile with in purple the improved Lennard-Jones and in green with the improved Lennard-Jones with the induction term. We can see that the inclusion of the induction term has considerably significant impact to the system because the water molecules tend to prefer interacting with each other after the introduction of induction potential. Also, we can see this phenomena in the right part in this graph, the radial distribution function, where you can see that the higher peak of the water molecules are located near a certain water molecules. So in the case of the improved Lennard-Jones plus induction. So in this case, we can say that the water-water interactions are unfavored with the improved Lennard-Jones and favored with the interactions water graphite. In the case of the water-water interactions are favored introducing the induction part. So at this point is important, is necessary to conclude that we need there is from a computational cost is considerable the impact of the total CPU time for the pure gas we have observed when we have introduced the induction potentials after small modifications in the code. So, but it's important the inclusion of this potential of this part because we have observed that the total potential energy were modified. So, to conclude, we can say that in the future molecular dynamics simulations, we want to quantify using a very long simulation time will be accessible for our system to describe better the part of improved Lennard-Jones potential, take care about the induction part that is necessary when we speak about water-water interactions. Okay, I have concluded. This is the acknowledgements, and obviously, thank you for your attention. So I don't know if there is some question from the classroom or from the online people. Yes.
Are you planning to include the polarizability as well?
So the parameters that introduced in the improved Lennard-Jones are from the polarizability, and this is the favor of this type of potential because in some cases are from the experimental, properly the experimental results. In other cases from the theoretical formulations, but including all polarizabilities. For the reason describe better that classical Lennard-Jones potential, how you know, okay? Thanks a lot. So I conclude the session. Thank you, the theoretical chemistry and computational modeling and application session. And I think so move for another session.
4 minutes.
4 minutes, okay. Sorry.
Live streaming is on.
Ok.
Siamo?
Sì.
Mi vedono?
Sì, sì.
Good morning to everybody. Welcome to the Compastro workshop dedicated to computational astrochemistry. Both gas phase approach and surface gradient approach will be considered in this workshop. The first part of the workshop is this morning, while the second part is in the afternoon after the keynote lecture. So we can start with the first speaker of the morning, which is Andrea Lombardi, and he will present the work titled the CH2CH2 plus OH gas phase reaction, formaldehyde and acetaldehyde formation routes. Please, Andrea.
Thank you.
Prima.
Just a second. Okay, good morning. Okay, my presentation here is about this reactive process in connection to possible formation routes of formaldehyde and acetaldehyde in the interstellar medium. Ok, quindi vado così. Okay, as probably you probably know if you participated to the previous workshop, in the interstellar medium is rich of molecules which are relatively complex in the astrochemical sense. Molecules made up by more than 6 atoms are considered to be complex, given the very, very peculiar conditions, environmental condition of deep space. And the presence of these molecules is very important for many reasons. The main reason astrochemists and astrophysicists are interested to the detailed knowledge of the molecules present in the interstellar medium is because these are indirectly a probe of the physical conditions of the environment. I mean, density, temperature, and many other aspects. In total, we have more than 200 molecules detected so far by different methods. These are of many types, essentially organic molecules like aldehydes, ketones, carboxylic acids, esters, amides, and so on. In addition, we have molecular ions, because ionizing photons can be present. We have radicals, of course, and we can observe the presence of unusual isotopes because the isotopic distribution can be different in space. And we have also many structural isomers which are normally not observed in our usual chemistry on the planet Earth. So what is the task for the astrochemists? It is to understand how the molecules which are proved to be there can be formed by reactions of many types. And in practice, we have to identify the process that contribute most to the formation of a given molecule. We have to collect all the possible processes, and we can use these processes to build up kinetic models of a given environment. And using these models, we can try to explain, to reproduce the abundance of the species which are observed. The difficult task is to study all these processes in the range of condition of interstellar medium, which are quite extreme conditions, in the sense that we have very, very, very cold temperatures in a range which can start from a few degrees up to 10, 50, or hundreds of degrees, and very, very low densities. And you know that in this condition it is difficult to design also real experiments, because to reproduce this condition is not always an easy task. Also, direct theoretical simulation of the processes that generates these molecules, such as collisions, direct simulation of collisions, is not so easy due to the extremely low kinetic energies, which means that we have to face a range of computational difficulties, and so on. But anyway, what we are forced to try to obtain, it is in practice the calculation of the rate constants for each process. We need the rate coefficients for any possible chemical route available for the system. We need to determine the temperature dependence of this rate constant in a certain range. Here there is a typical fitting function, which is a modified Arrhenius function. But also we have to identify the nature of the reaction products, because we don't know in advance which are the products that really can be formed. And there are some rules that generally apply in the interstellar medium. So the favorite processes are those which are exothermic, in a sense. Of course, due to the extremely low kinetic energy, we look to barrierless processes. Otherwise, the probability, the reactive probability, it is expected to be very low. And also there is the necessity, it is necessary to consider the possible outcomes of any reactive process, because more product channel could be principal accessible, so we have to determine the branching ratio of these possible channels. And also we have to find the reactive pathways, because when we make collide, we take close by two different molecules, many possible reactive pathways in alternative are possible on the full-dimensional potential energies of this. Why we are interested in this reaction? We are interested in this reaction because formaldehyde and acetaldehyde are two possible products of this ethylene plus OH reaction. Particularly, acetaldehyde, it is very interesting in view of this potential prebiotic role, because it could be, in principle, a precursor of carbohydrates and acrolein, for example. And to establish that this process can generate in a certain not negligible amount acetaldehyde, it is an interesting subject of study. So acetaldehyde has been studied for many years, since many years. Here there is a scheme of the possible gas phase processes that can, in principle, generate acetaldehyde. I mentioned gas phase processes because also mechanisms based on the interaction of the molecules with the dust grains are in principle possible and have been studied. We are considering the gas phase processes, and we want to add to this scheme our reaction, ethylene plus the hydroxyl radical. And what we know, just to start to consider this process, so we know that there are two exothermic channels, one leading to formation of formaldehyde, and the other one leading to formation of acetaldehyde. So these two channels are those that in principle have to compete when these molecules can react in the interstellar medium. We know something about this kind of processes. This is a radical plus neutral molecule process. In general, experiments indicate that this kind of processes exhibit a negative temperature dependence, in the sense that the reactivity increases as the temperature decreases. This is the opposite of the standard Arrhenius trend, but it is quite common for these kind of reactions. We should try to understand why this behavior takes place, and we also should try to evaluate the branching ratios for these two channels. It means how many formaldehyde is formed and how many acetaldehyde is formed when these two channels are set in competition. The methodology is similar to that Emilia Aragao was illustrating in his talk in the previous section. We want to we need an interaction potential. We know that this the first step of these reactive processes, it is the formation of a weakly bound complex. The formation of this kind of dimer, actually, it is thought to be driven by intermolecular interaction. So we need to, at first place, to obtain a reliable intermolecular interaction potential. And this has been done representing the interaction as the sum of two contributions. One, the ILJ, it is a van der Waals type interaction, and the second term accounts for the electrostatic interaction. Of course, this second term is generated by a charge distribution, atomic charge distribution, that we have to provide to calculate the Coulombic interaction term, as it is shown here in the slides. These are the parameters that we determined for this system. We have equilibrium distance and well depth for the atom-atom interactive interaction pairs. We also have atomic charges for carbon, hydrogens, oxygen, and for the molecules and the radicals. And this is the potential profiles that we can obtain applying this semi-empirical approach, letting the system, the two molecules, to get close along along a line, okay, which connects the center of mass of the molecule with the hydrogen atom of the radical. This is the characterization of the reaction pathways. We can see that there are, as I anticipated, two exothermic reactive channels, formaldehyde and acetaldehydes, and this is the sequence of intermediate stable configuration and transition states that leads to the asymptotic product channels. As you can see here, the first intermediate stable configuration corresponds to the dimer, to the wiki-bound dimer, which is actually represented here. And the second step to the transition states is the formation of a very deep second intermediate. Then there is a sequence of other transition states at intermediate, up to reaching the reactive, the final products. This is the detail of the structures that have been calculated for the intermediates and the products, and for the transition states. Okay. And here in this table, you can see the enthalpies variations and the barrier heights for the various steps which lead to the formation of products. Kinetic calculations can be performed having this information, the previous information. Of course, we need to we want to represent the formation of this first dimer using the two-body capture theory, okay, which is a simple theory that it's useful to calculate rate constants for this kind of processes. This is only the first step of the sequence of steps. Then we need the rate coefficient, which can be obtained with the well-known RRKM theory in this way, using the number of states up to energy E and density of states. Then, once we have all the necessary rate constant, we can set up a master equation. Solving this master equation, we can get, of course, a rate constant for the formation of products and also branching ratio. These are actually the branching ratios that we obtained applying this master equation approach at different temperatures. And we can see that, of course, the formation of formaldehyde is predominant, okay, because at least at low temperature, there are a few order of magnitudes if we compare formation of formaldehydes and acetaldehydes. These are numbers indicating these are percents, because most of the products ends up in the dissociation of the wiki-bound complex. So most of the collisions leads to dissociation. Some of a small percentage of these colliding events leads to formation of products, and this is the absolute. This is the percentage for the two alternative channels. We do not report here rate constant because we found some disagreement between existing theoretical calculation of this for this kind of process. Although these were not directly comparable, but anyway. And this is probably due to the bad performance of the capture theory that we applied to obtain the first rate for the first step, which is the formation of the weakly bound complex. So we need probably to use a more accurate and less approximate approach to obtain the K1, the first rate constant, and this will lead probably to better results. So thank you for your attention, and I can finish here. Okay.
Thank you, Andrea, for this nice and interesting presentation. We have just 1 minute for questions. Are there any questions also from remote? I have a curiosity.
Yes.
So it seems that for long-range interactions, semi-empirical approaches are better than ab initio calculations.
Yes, apparently yes, because at least you can get a reliable potential profile. Instead, sometimes ab initio calculations are not really feasible for this kind of systems. So you're not able to obtain full accurate and convergent results. So in this case, it's probably better to rely on the semi-empirical approach. And this is, in a sense, we have seen in previous talks that this is a quite common situation. So one does quantum chemical calculation without having the guide of semi-empirical method.
Okay, thank you very much. Are there other questions? Okay, so we can move to the next speaker since we are a little bit late. Next speaker is Stefano Falcinelli, from remote.
Okay.
Stefano Falcinelli from the University of Perugia, which will is connected from remote. Stefano, are you there?
Yes, Marzio, thanks.
Okay. And Stefano Falcinelli will make a presentation titled Free Methane from the Ionosphere of Mars towards a prototype Metallation reactor, a project producing fuels via plasma-assisted carbon dioxide hydrogenation. Stefano.
Okay, thanks, Marzio. I will try to share my desktop, the presentation. Can you see?
Not yet.
No.
Not yet.
Not yet.
Can you see now?
No.
No.
No? Oh,
In the bar above, it's the third button.
yes. Okay. I'm clicking there in the desktop sharing, but I cannot obtain nothing. I don't know why.
What's the problem? Sorry.
I cannot. I'm clicking on the link, but to share the desktop, but it don't works. I cannot do nothing.
Try to leave the room and enter again on Jitsi.
Okay, I'm.
We'll wait for you. Okay.
Okay, thanks.
You're welcome. Okay, let's wait 30 seconds. And then I try to help Jessica.
Yeah.
Jessica, can you hear us? If you are trying to talk, you are muted. So you need to unmute your microphone. Now close YouTube. You have to go in the link of the Jitsi room and try to close YouTube. And try to leave the room and enter again here on Jitsi.
You means that I must do that?
I was talking with another presenter.
Ah, okay. You are okay. You are speaking with another people. Okay.
Yeah.
Thank you.
Because she's having problems. Professor Falcinelli, if you want to try again.
Yeah, I'm trying. I don't see no changement.
No, so it's not working, the sharing button.
Yeah.
Which browser are you using?
Oh.
Try with Chrome, it's the better one.
Okay, I can try to exit and changing the, because now I'm using Firefox.
No, try with Chrome.
Okay, I'm sorry, but
no, don't
I'm
worry.
trying to. Okay, I will do that.
Thank
okay.
you. Okay, let's move to Jessica. But can you hear us, Jessica, right? Have you tried to get out of the room and enter again?
Is on.
Okay, I will try to share my desktop. Okay.
Okay.
Okay. Do you see now?
No.
No. Oh.
Yes.
If you want, you can send the PDF to me and I will share it for you.
Okay.
That's okay. Jessica, you are unmuted. Try to talk.
No, it is not working.
No, I try to exit it now. For
No.
Jessica Pereira, I think it's a problem of your connection.
No,
No,
something is.
no, you can try to send to me your presentation.
Okay. What is your address?
Yeah, I'll give you in private here.
Okay.
Yes, Jessica, you are unmuted, but we are not listening to you. Have you tried to check, Jessica, the settings of Jitsi? In the three dots, go to settings and look which microphone Jitsi are you trying to use? Okay, Professor Colcinelli, I give you my address. Can you see it?
Yes.
Okay, I'm waiting for the presentation.
Okay. Marzio? Sì, l'ho mandata. Ora non so. Did you receive the presentation or not?
Not yet. But Professor Rosie is trying to help me. Just 1 second.
Okay.
Okay, we have the presentation, Professor Falcinelli, so I will help you with that. Can you see our screen? Professor Falcinelli?
Yes.
Ok. No, è uscito. Ok, Professor Falcinelli, maybe I lost the Wi-Fi. Okay, we are waiting for Professor Falcinelli. He's having technical problems.
Is on.
Okay. Can you listen me?
Yes. Okay.
Oh, good.
You may start.
Okay. I don't see nothing in my desktop. Okay. You can see the presentation?
Yes, we can see the.
Okay. Okay. Good. Good. Okay. I'm sorry for.
Professor Falcinelli, are you seeing the presentation here on Jitsi?
No, I cannot see nothing.
Nothing?
I see just black. All is black. Okay, but it's okay. You can please go on with the slide. Maybe I can tell you when you can go on. Okay.
Okay, because we can see the presentation.
I cannot see nothing.
I think it's a problem of your connection because I'm seeing on YouTube your presentation.
Okay, so we can.
But okay, we can go. We can start.
Okay. Thanks for giving me the opportunity to present my contribution today. I'm sorry for this inconvenience, and my talk concerns an experimental and characterization of the best working conditions of a metallation reactor. It was specially designed and built in order to convert waste carbon dioxide into methane using renewable energies. In the first slide and the second slide, you can see a picture of this device. The used chemical process is reported in the second slide, and it is the well-known Sabatier reaction, which allow the hydrogenation of carbon dioxide into methane and water by the use of a nickel or ruthenium metal catalyst. First of all, we determined the best working condition for our methanation reactor, and you can see in the third slide the experimental determination of the methanation yield. We obtained the best methanation yield of about 84% when we use a reagent mixtures, carbon dioxide and hydrogen mixed in a ratio of 1 to 5. Second, after that, we determine also the best pressure inside the reactor, the total pressure, and the best condition was determined as 2 bars. Okay, so on we can after this first determination, we decided to try to perform a methanation process without the use of the solid catalyst. For such a purpose, we try to use a plasma-assisted catalysis. Since our research group is able to produce various kind of plasmas, and we can do that using different gaseous mixtures of our interest, and particularly we determined the production of various plasmas using different carbon dioxide hydrogen mixtures. We can do that using both radio frequency and microwave discharges that are operative in our crossed molecular beam apparatus that is reported in the 5 slide. After this determination, after the production of this kind of plasmas, we were able also to characterize the produced plasmas. In the sixth slide, you can see that it's possible for us to obtain a dissociation of carbon dioxide in our generated plasmas of about 62%. This means that the plasma generation is a good strategy in order to allow carbon dioxide to react in the subsequent hydrogenation process that is of our interest. In order to allow
Hey, sorry, Jessica, we can see you and hear you. Can you please mute yourself? Thanks.
I cannot. Can you hear me?
Yes, Professor Facinelli, go on.
I'm listening other voices. I kind of okay. I go on. Okay. You can go to the next slide, and I can show you that after the generation of this kind of plasmas, we were able also to characterize chemically the produced plasmas by using mass spectrometry technique. I reported in this slide two mass spectra for the plasmas that was generated by using a microwave discharge plasma source with two different gaseous mixtures where carbon dioxide and hydrogen are mixed in a ratio of 1 to 3 and 1 to 5. You can see from these mass spectra that it's possible to convert carbon dioxide into simple hydrocarbons or simple organic molecules such as methane, formic acid, and for example, dimethyl ether, that are able to be used as fuels. This means that with the plasma generation is possible in principle to avoid the use of the metal catalyst, and we can convert a waste substance as carbon dioxide in useful species. I just want to stop my presentation, and just I would like to thank the company and the institution that are working with us in this project. They are reported in the next slide, and obviously I thank all of you for your patience and your attention. Thank you very much. Okay.
Okay, Professor. Now it's the turn of Jessica. Yes, we are finished. It is finished. The questions?
Are there any questions? I think that
From the audience?
people can still hear us. It seems that we have big connection problems today. Sorry to wait with everybody. Stefano, can you hear us?
Sorry, Matteo. Can you repeat?
Because it seems that we have a connection problem.
Yes, sure. Okay, thank you very much. I'm sorry for
no, thank you.
This very bad presentation. I'm sorry. This is my fault.
Don't worry.
Okay, thanks.
Anyway, we have time for any questions, also from home, any curiosity. I just a question. Which are possible practical applications of this interesting study? You already mentioned some connections with industries that you have.
Yes, Marzio. We have a connection with several industry companies. First of all, with the PLC company located in Cerra, Naples. That was the company that was put a lot of money to realize this methanation reactor. After that, we are collaborating with a company that provide biogas produced from fermentation of exhaust vegetables, and this biogas will be converted totally in carbon in methane in the next month in our methanation reactor, in order to demonstrate that it's possible to work in a circular economy strategy for a company that are interested to use our methanation reactor.
Okay, thank you very much, Stefano.
Thanks to you. Thanks.
And sorry for the connection problem. Okay, we can move to next speaker, which is Jessica
Ferrero. Yes.
She's connected now.
Yeah.
Yes. And Jessica will talk about make a presentation titled "Ab initio calculation of binding energies of interstellar sulfur-containing species on crystalline water ice models." Please, Jessica, you can start. Sure.
Can you see my screen?
Yes, Jessica.
Okay, perfect. So, good morning, everyone. First of all, I would like to apologize for the inconvenience for the connection. And if it may be of any use, I discovered that it was an internet connection problem, meaning that the university line wasn't letting me share anything or connect. So with that said, let's talk about the binding energies. So in the solar-like planetary system formation, we can divide this process in 5 phases. And I am going to focus on the first one, the Prostellar Phase. The material in this environment is agglomerated in clouds that are divided in diffuse and dense clouds based on their physical properties. And we will focus on dense clouds where the density is higher, and this allows the presence of not only atoms and small molecules in the gas phase, but also of complex organic molecules. And the temperature are lower, around also sometimes 10 Kelvin, so that the dust grains are not only bare, but they are covered with several layers of water-dominated ices. And here in this picture, you see also the presence of other small molecules. This is due to the phenomenon of absorption that take places at this low temperature. And from an energetical point of view, the absorption is described by the binding energy. So it is the energy of absorption of a chemical species on a surface. And we are interested in physisorption, so no chemical reaction occurs in this event. But why binding energies are so important? Because in the numerical models that describe the evolution of these environments, the binding energies are essential parameters. And as you see, the rate constant that describe the accretion, the diffusion, and the desorption of species onto the grain are proportional not only to the binding energy, but to the exponential of a binding energy. So that a small variation in this parameter will have a huge impact on the model, and therefore the chemical composition will change a lot. We decided to compute the binding energy of 8 sulfur-bearing species, and as a model of ice, we chose a crystalline model. We know that the majority of the ices in the interstellar medium are amorphous, even though there is evidence for the presence of some crystalline ice in protostellar disk and in protoplanetary disk and in shocks. But we chose a crystalline structure because from a computational point of view, it's easier to model since it is a known structure, while we don't have any sure model of an amorphous ice. And it is also computationally cheap because thanks to the symmetry, the model will be smaller and therefore it will occupy less time to compute. So this is the 010 surface of our proton-ordered crystalline ice that we use for the computation of binding energies. And these dangling hydrogens and oxygen on the surface are the binding sites, and they are also responsible to the dipole of the surface. So for our ab initio periodic calculation, we need two ingredients. One is the DFT functional, so we chose the B3 Lip3 with the dispersion D3 from Grimm in the Becker-Johnson damping scheme, and the Ulrichs triple set equality supplemented with a double set of polarization function basis set. So the binding energy is a positive number because it is the opposite of an interaction energy. And in few words, our interaction energy is corrected for the basis set superposition error. When we also correct for the 0-point energy, we will then talk about adsorption, enthalpy adsorption. But this is not the focus of this talk. So after computing, we could say that our DFT results are accurate but expensive. And why am I using these two adjectives? Accurate because after performing our calculation, we decided to refine the energies at couple cluster level. So we cut a small cluster in orange from our periodic model, and every time the cluster was made by the adsorbed molecule plus the two most interacting water molecules, on which we computed both the DFT binding energy and the couple cluster binding energy at a complete basis set limit using the June correlation consistent basis set. So we computed this difference in the formula, and we added to our DFT results previously computed on the periodic model in order to obtain the onium-corrected binding energies. And when plotting these binding energies against the full DFT values, we see that the correlation is very good, and therefore this is why we can assess that our results are accurate. But also they are expensive, because as you may imagine, the DFT calculation on big models will require a lot of time. And in provision to apply this procedure to more molecules and bigger ice models, we need to find a cheaper methodology. And this is why we are using the HF3C method by Scher and Grimm. First of all, we used it both for the optimization of geometry and the calculation of the energy, which resulted in a very poor correlation against always our full DFT results. So this is why we decided to try to refine the energy with a single point calculation at DFT level, namely B3 Lip. And in this case, when we do this procedure, we see that the correlation between the results obtained in this way are much more in agreement with the full DFT ones. And this is the reason why we will apply this methodology in future for the calculation of other binding energies. So how our data are entered the panorama of the other literature data. So here I'm doing a comparison with other two computational work, one by Weckelam and others, 2017, in which the ice was simulated with just one water molecule, and the other one from Das et al., 2018, in which the ice model was simulated by a water tetramer. You can see that our results, the blue rhombi, are in any case higher than the previous data from the literature. And this may happen for two reasons. One is the different level of theory. So Weckelam and Das used other functionals and other basis set to compute their binding energies, but also they did not correct for the BSSE, which we did. But also, and probably the huge responsible for this difference is the ice model, because you have to keep in mind that in the ice, the hydrogen bond network has a cooperative effect, which is very strong and enhances the binding, the strength of the binding sites a lot. And this results in stronger binding energies, especially when we are treating polar molecules. So in the future, we aim to enlarge the set of binding energies, computing other sulfur-bearing species, which are in this case open shell and not more closed shell like the previous ones. And also we aim to compute the binding energies on the amorphous ice model, so that we will provide 8 binding energies for each molecule and therefore create a distribution, which is more representative of the reality of the ice, since we don't have a periodic ice on our dust grains, but we have an amorphous model with different binding sites, and they can be better described by an amorphous model. So in the end, we computed the binding energies of 8 sulfur-bearing species, of which OCS resulted to be the one with the lowest binding energies, while SO2 is the one with the highest binding energy. Couple cluster refinement confirmed the quality of our DFT results, and the DFT HF3C procedure turned out to be a good procedure to enhance the number of binding energies calculated and finally defined distribution of binding energies. Thank you for your attention.
Thank you, Jessica, for this nice and interesting presentation.
Thanks.
We have time for questions or curiosities. Just a curiosity. You use also an nonium approach in your study.
Exactly.
Do you think that this can be a valuable approach for to study systems like those you consider here? You think to use it also in the future? Or
Well, when calculating on the crystalline ice model, sure, it is more feasible to being able to cut a cluster, because the problems are the dimensions of the system. Of course, we cannot apply couple cluster to periodic calculation, but the problem would be also with a great number of atoms. So I think that if we can prove that our results with DFT are good enough compared to couple cluster, then we just can rely on the DFT procedure and keep computing on larger models, because it would be a very time-consuming work, and it is not feasible if you want to compute a large number of binding energies.
Okay, thank you, Jessica. Thank you again for your interesting work. So we can move to the next speaker, the last speaker of the morning. The last presentation will be presented by myself. It's a computational study on the attack of nitrogen and oxygen atoms to toluene.
Okay, we need 30 seconds for organize everything. We can start?
Yes. Okay. Everybody can see the screen, I hope. So this is a theoretical study on the interaction between nitrogen and oxygen atoms with toluene. And this is a collaboration between our research group of Perugia, the beautiful city here shown, and the Polytechnic of Milan. The synthesis of nitrogen-containing organic molecules is a crucial step in prebiotic chemistry, as they are potential precursors of important biological molecules such as the amino acids. In this respect, the interaction of nitrogen in its excited double-D state with small hydrocarbons is particularly interesting, because nitrogen in this excited state is particularly reactive. Interestingly, simple species belonging to both nitrile and imine families have been detected in the interstellar medium and in the upper atmosphere of Titan. Titan is the massive moon of Saturn and is characterized by a surface temperature very low and by an atmosphere composed mainly by molecular nitrogen and small hydrocarbons like methane. This atmosphere is particularly interesting since it is believed to be somewhat reminiscent of the primordial atmosphere of Earth. The molecular nitrogen with methane, thanks to the energy provided by sunlight or by energetic particles, can react and give rise to more complex molecules like benzene, organic molecules, and at the end, they can give rise to toluenes, that is, very complex organic molecules. However, molecular nitrogen is characterized by a strong bond and is difficult to chemically fix in compounds. However, the observation of nitriles in trace amounts indicates that some forms of active nitrogen are produced by several processes, for instance, electron impact or absorption of ultraviolet light. This energy can promote the nitrogen from its ground state to some excited states, and some of them can be dissociative, giving rise to atomic nitrogen, not only in the 4s ground state, but also in excited states, in particular in the doublet D excited state, which is very interesting since it is metastable, but it has a very long radiative lifetime, around 48 hours. This state is particularly reactive and is able to, nitrogen in this state can react with closed shell molecules. It is interesting to notice also that in the upper atmosphere of Titan, the presence of nitrogen in the excited doublet D state is mainly at an altitude which is the same where we can find a large amount of benzene and phenyl radical, which can easily react with methyl, giving rise to toluene. The interaction of nitrogen in its excited state with small hydrocarbons has been already investigated in our laboratory, both from an experimental point of view through the cross-beam technique and from a theoretical point of view. Now we want to extend this study to the interaction of nitrogen in this excited doublet D state with aromatic hydrocarbons, in particular with benzene and toluene. But toluene is important also for the reactions with atomic oxygen, with oxygen in its 3p ground state, because these reactions are very relevant for combustion and atmospheric chemistry. And these reactions have a crucial role in full oxidation to the production of also polycyclic aromatic hydrocarbons. So we started also a synergistic experimental theoretical investigation of the reaction between toluene and oxygen in the 3p ground state. So our approach is essentially this one. We perform in our laboratory in Perugia the experiment using the cross-molecular beam technique in order to obtain information on the branching fractions, branching ratios of the investigated elementary reactions. Through theoretical calculations, we study the potential energy surfaces of interest, and then perform kinetic calculations. We obtain information about the branching fractions and the rate coefficient. At the end, we can use all these informations in order to improve the modeling of combustion, plasma, atmosphere astrochemistry, so we can have applications in many relevant fields. The theoretical calculations were performed considering the density functional theory approach for geometry optimizations and for the localization transition states. We use for these studies two different functionals, the omega B97 XD and the B3LIP functional, with both more limited basis, Popper basis set, the 6311 plus GDP, and a more extended triple zeta basis set, the augmented CC correlation consistent BTZ Dunning basis set. The nature of the transition state has been characterized performing intrinsic reactive calculations, coordinate calculations, and then the energy were refined performing couple cluster calculations for all the stationary points. It is interesting when we consider interaction of toluene with nitrogen in its excited WD state and oxygen, both in the 3p ground state and in the excited 1d state. It is interesting to notice the interaction, the geometry of the minima. For nitrogen, first line in this slide, we can see that nitrogen in this excited WD state, the nitrogen is able to break a carbon-carbon pi bond, and it gives rise to the formation of two sigma bonds with two different carbons. The same situation is observed for the oxygen in the ground triplet P state, last line of the slide, while the oxygen in the first excited singlet D state, middle line, is able only to interact with a carbon atom. Is not able to interact simultaneously to two different carbon atoms. This is quite obvious, of course, because in the oxygen, in its ground state, triplet P ground state, we have three electrons all with the same spin, and so only one of these electrons is able to interact with the electrons involved in the pi bond with two carbon atoms, which have, of course, opposite spin, show opposite spin. The main products we obtain from the reaction between oxygen in its ground state and toluene, as is confirmed by the experiments, are methyl and hydrogen. So we have these two proton channels, the formation of methyl and the formation of the hydrogen. We can notice that in both cases we have an initial barrier which lies above the reactants. So these reactions can be important in combustion chemistry, but of course not in the interstellar medium where we do not have much energy available. It is interesting also to notice the geometry of the transition state leading to the loss of methyl or hydrogen. In both cases, the leaving group leaves the molecule in a direction perpendicular to the plane of the molecule, and this is observed also experimentally. The methyl and hydrogen loss are important channels also in the case of the reaction between nitrogen in the double D excited states and toluene. In this case, we can notice that we do not have any barrier lying above the reactants. So these reactions are, of course, important also in the interstellar medium or in environment like the Titan atmosphere. To conclude, so we can say that the main channel for the reaction between oxygen and toluene are those leading to the hydrogen and methyl loss. But we have also observed experimentally an important channel leading to the formation of carbon monoxide. Of course, this cannot happen in the triplet ground state surface, but should happen in the excited singlet surface. So for this instance, in order to complete our study, it will be necessary to consider also the intersystem crossing between these two surfaces. For the reaction between nitrogen, WD, and toluene, it is important also the formation of novel carbon-nitrogen bonds. In particular, we observe the formation also of hydrogen cyanide and similar compounds. Thank you very much for your kind attention, and this is the funds provided for this study. Thank you to everybody. We are a little bit late, but if there is any question, curiosity, I'm here. Although I think that it's time for lunch, so I thank again all the speakers of the morning, and we will go on with our workshop in the afternoon after the keynote lecture. Thanks again to everybody.